Today we will talk about the citric acid cycle. I am Yan Lee, who will be starting this video, and below are the names of people who will also be presenting. This is the table of contents. Before we take a deep dive into the details, let's look at the names of citric acid cycle. The citric acid cycle has three different names. It is also called the TCA cycle or the Krebs cycle. First, it is called the citric acid cycle because citric acid is the first product of this cycle. This citric acid is formed by the concentration of acetyl-CoA with oxaloacetic acid and water. It is also called the TCA cycle or the tricarboxylic acid cycle because citric acid has three carboxyl groups. Lastly, it is also known as the Krebs cycle because Sir Hans Krebs is the first person to discover this cycle.
So why this TCA cycle is so important? The importance of the TCA cycle is evident in two main aspects. First, this cycle significantly increases the efficiency of energy production. Through a series of processes, the generation of three molecules of NADH and one of FADH2 provides high energy electrons that are converted into ATP via the electron transport chain. The creation of these high-energy molecules is essential for supplying the energy needed by our bodies. Second, the TCA cycle acts as a metabolic hub by producing intermediates that encompass various nutrients. These intermediates transform not only glucose but also amino acids and fatty acids into usable forms, contributing to the synthesis of various components that make up our bodies. The presence of these intermediates means that the TCA cycle does more than just generating energy. It plays a central role in regulating and integrating various metabolic pathways within the cell. We told about the importance of TCA cycle, and when we see the complicated and cyclic structures of TCA cycle, we can understand more about this importance. TCA cycle produces a variety of intermediates. These intermediates do not merely circulate within the cycle to produce energy. They also flow into other metabolic pathways to perform various biochemical functions. For example, citrate provides acetyl-CoA necessary for fatty acid synthesis, while alpha-ketoglutarate plays a crucial role in the synthesis of amino acids. The presence and utilization of these intermediates allow the TCA cycle to function not just as a simple energy-producing mechanism, but as a central hub for multiple metabolic pathways within the cell. The cyclic structure of the TCA cycle allows for the continuous recycling of intermediates while efficiently generating energy. This structure enables optimized energy extraction at each step and simultaneously allows the generated NADH and FADH2 to produce more ATP through the electron transport chain. Moreover, this cyclic structure incorporates regulatory mechanisms that allow the cell to flexibly respond to changes in energy demands. These aspects demonstrate that TCA cycle designed very sensitively to support both increased cellular energy efficiency and a central hub role in metabolic pathways. Now, let's go deeper to the TCA cycle. After glycolysis, we can get pyruvate, and pyruvate converts to acetyl-CoA. This acetyl-CoA has high energy bonding. We have to use this energy via TCA cycle. First, acetyl-CoA, which consists of two carbons, combines with oxaloacetate which has 4 carbon, and produces citrate, which has 6 carbon. This reaction is irreversible, which means it can't go backward. The enzyme called citrate synthesis mediates this pathway. And next, the enzyme called aconitase mediates citrate turning to isocitrate, and there is no change in carbon number. This is a reversible reaction. These two reactions are important, although these steps do not directly produce energy. They are crucial because they initiate the metabolic cycle that is essential for later energy production. These steps also generate important metabolic intermediates that are used in subsequent reactions. Moreover, they regulate the flow and activation of the entire cycle, ensuring efficient energy and biomolecule synthesis throughout the cell.
I am Yan Lee again, and I'll be explaining Step 3 and Step 4 of the cycle. Step 3 is an oxidative decarboxylation step, which the isocitrate is converted into alpha-ketoglutarate by an enzyme called isocitrate dehydrogenase. During this step, isocitrate is oxidized into oxalosuccinate and NADH is formed. Then, the oxalosuccinate undergoes decarboxylation step and carbon dioxide is formed. To sum up everything, isocitrate with 6 carbon is turned into alpha-ketoglutarate with 5 carbons, and this is done by the enzyme called isocitrate dehydrogenase. During this process, the carbon-carbon bond is broken, and NADH is formed, and carboxyl group is released as carbon dioxide. This step is irreversible because the CO2 is so stable, they do not want to go back to the previous form. Also, the allosteric enzyme isocitrate dehydrogenase contains allosteric regulatory sites and activator molecule ADP. When the cell needs to produce more energy, ADP will bind to the regulatory sites, and then affinity for isocitrate increases, causing the rate of total cycle to also increase. However, when we don't need more energy, ATP or NADH acts as allosteric inhibitors of isocitrate dehydrogenase, and the rate of the cycle decreases. The fourth step is another oxidative decarboxylation step in the cycle. Carbon-carbon bond is again broken, and the carboxyl group is released in the form of carbon dioxide. In this step, alpha-ketoglutarate over here, which has 5 carbon, is converted to succinyl-CoA over here of 4 carbons. This is done by allosteric enzyme alpha-ketoglutarate dehydrogenase complex. During the process, energy molecule NADH is again formed. Also, because oxidative decarboxylation is thermodynamically favorable and CO2 is so stable, this reaction is irreversible. The fourth step also plays a role in regulating the cycle. When energy is high in the cell, ATP, succinyl-CoA, and NADH all play a role of allosteric inhibitors and decreases the rate of this cycle.
I'm Hanako and we'll talk about Step 5 and Step 6 of TCA cycle. Let's start with Step 5. It is conversion of succinyl-CoA to succinate. Succinyl-CoA synthetase is the key intermediate of this step. The number of carbon is 4, because 1 carbon dioxide molecule was released during Step 4. Energy is produced in this reaction. This is a reversible reaction because there are no enzymes reversing this reaction. Conversion occurs in mitochondrial matrix where succinyl-CoA is located, and this is equation of the reaction. Succinyl group from succinyl-CoA is transferred to GDP, forming succinate and GTP, while coenzyme A is released. This reaction involves substrate-level phosphorylation, where the high-energy thioester bond in succinyl-CoA is utilized to phosphorylate GDP and drive the formation of succinate. Next step in the TCA cycle involves the conversion of succinate to fumarate catalyzed by the enzyme succinate dehydrogenase, also known as complex 2 of the ETC. There is no change of number of carbons, so 4 carbons are remained. This reversible reaction occurs in the mitochondrial matrix, where SDH is located. And here, SDH stands for succinate dehydrogenase. During this process, succinate is oxidized by SDH, leading to the release of 2 high-energy electrons to FAD, forming FADH2. While no change in the number of carbon atoms occur, this step is pivotal for both the citric acid cycle and the electron transport change. Following the conversion to fumarate, SDH can be recycled to participate in subsequent cycles of the citric acid cycle.
Hello, my name is Isak Chung, and I'm going to explain remaining citric acid cycle reactions step 7 and step 8. The step 7 is fumarate to malate reaction, which requires the fumarase enzyme. This fumarase enzyme adds a water molecule called a hydration reaction, which is adding a hydroxyl group and a hydrogen ion across the carbon-carbon double bond to form malate. The carbon number of malate is 4. This reaction is irreversible because, as I mentioned earlier, fumarate has a double bond, which is the energy state is high, and it does not generate energy separately. It is not a reaction regulated by enzyme activity. Consequently, this adding water reaction can occur without extra energy input. It means it does not affect the overall oxidation state. The step 8 is malate to oxaloacetate reaction, which requires the malate dehydrogenase enzyme. This malate dehydrogenase enzyme catalyzes the oxidation of malate to oxaloacetate using reduction of NAD+ to NADH. Oxaloacetate has 4 carbon, and this reaction is reversible, and energy molecule NADH is produced. Also, malate dehydrogenase enzyme activity is not controlled. As I mentioned earlier, this reaction is dehydrogenase reaction, which is oxidation. So this reaction releasing energy, and this reaction is thermodynamically unfavorable because it wants to get energy. So they lose 2 protons through oxidation, but they generate the double bond. Then, how this reaction occur? The answer is by citrate synthase. It conducts the reaction, and so oxaloacetate concentration keep lower and pulls the reaction forward. Consequently, this reaction can occur in our body. The second part is about beriberi disease. Do you heard about the beriberi disease? I will explain more detail in next slide. The beriberi disease is known as a thiamine deficiency. The thiamine is thiamine B1. This thiamine deficiency is caused by eating Polish rice or cassava root without much if any thiamine-containing animal products or vegetable. Also, have elevated levels of blood pyruvate and alpha-ketoglutarate, especially after consuming a meal rich in glucose. In next page, I will explain how thiamine deficiency causes this disease and relationship with citric acid cycle. As we can see in left picture, the thiamine is required for the synthesis of TPP, which is thiamine pyrophosphate. And this TPP is a prosthetic group in the pyruvate dehydrogenase complex and alpha-ketoglutarate dehydrogenase complex. So, thiamine deficiency reduces the activity of these enzyme complexes and causes accumulation of pyruvate and alpha-ketoglutarate. Namely, the citric acid cycle is not work proper, so our body cannot make ATP well. Thus, thiamine deficiency is caused this disease, and this disease is related to citric acid cycle.
Let's see symptoms of beriberi disease. There are various symptoms depending on the type of the disease. We can divide this disease into two big categories. They are dry beriberi and wet beriberi. Loss of sensation in hands and feet, loss of muscle function or paralysis of the lower legs, speech difficulties, nystagmus can be occurred as symptoms of dry beriberi, and increased hair rate, shortness of breath with activity, swelling of the lower legs is symptom of wet beriberi. And last, let's see how can we cure this disease. The most effective treatment of beriberi is to supplement thiamine. Thiamine is vitamin B1 we know very well. To prevent or treat this disease, we should consume thiamine-rich food such as grains, lean meats, nuts, and legumes. And this is end of our presentation. This is evidence of our activity. And last, this is role of each of us in this presentation. Thank you.